welcome to all of you to this seminar. I think most of you, or nearly everybody, knows me, but in case you don't, I'm Usama Tawil, the UNAIDS Country Director. I thank you all for taking the time and coming this afternoon, and would like to particularly acknowledge the presence of researchers, of civil society partners, of parliamentarians, of government actors, particularly the Department of AIDS Control, UN colleagues, as well as others who have come, and also our colleagues from the media. We have a special opportunity this afternoon of having the presence of Professor Sharon Lewin, who is with us from Australia. Professor Sharon Lewin is an internationally renowned researcher and also co-chair of the AIDS 2014 conference in Melbourne, which will be held on 20 to 25 July this year, 2014. As you can see from the title of this session, the focus is on a cure for HIV, Dare to Dream. This is a unique opportunity for the research and AIDS community in India to have and hear the developments that are going on in research, and hopefully she will have a presentation this afternoon, and in the dialogue afterwards you'll get an opportunity to ask questions and to dialogue with her on this. The eminent His Excellency the Minister of Transport, Roads and Highway, and the President of the Forum of HIV/AIDS. He has been Oscar Fernandez has been a key actor in the AIDS response for 10 to 15 years, and we have the opportunity of having him here. He never misses an opportunity actually to grace us with his presence and also give us his wise words with regard to how to take the AIDS response forward. We also have the opportunity of having Dr. Subbaraj, the new Secretary of the Department of AIDS Control, Ministry of Health and Family Welfare of the Government of India. We also have Dr. Ramesh Paranjapi, who's with us today. He's the Director of National AIDS Research Institute in Pune, and NARI is a leading research institute under the Indian Council of Medical Research and Department of Health Research. Lastly, it's also my honor to introduce Mr. Wayne Lewis, who's the Commissioner to India for the Government of Victoria, Australia. So just to go briefly through the program this afternoon, after my short welcome note, which I'm doing now, we will ask Dr. Subbaraj to say a few words as an introduction, inauguration also of this program. We will have a keynote address by Professor Sharon Lewin, followed by a discussion that will be facilitated by Dr. Ramesh Paranjapi, where you can take time to ask questions and so forth. And in closing, we have an address by Mr. Wayne Lewis, as I mentioned, Victorian Commissioner to India.
A cure for HIV. We have all assembled here to listen to Madam Saran for your important research, to hear as to what she is doing in this particular field. As all of us are aware, the humanity has been facing problems from various organisms from time immemorial. Our fight with the diseases is always on, never ending. One disease comes, one disease we are able to control, another disease comes. So this fight, I think this will continue as long as human race is there in the earth. We have been subjected to all types of tortures by various organisms. And virus, especially, virus has been in the forefront, causing a large number of diseases to the human society. This particular disease, HIV, all of us are aware of the implications. It has killed more than 25 million people in the world. such a huge devastation. But for the timely intervention of all agencies, but for the grant that we get, but for the technical support that we get, but for the protocols that we have received, I think this 25 million would have become much more. Today 25 million people died, and year after year people are also dying. But at least now that fear is not there. That fear that HIV will cause instant death, that fear is not there in the society. Today we have got very good ART system. I think that is the system that is available throughout the world to see that the disease is kept under control to a great extent. Today we find people living with the ART for 20 years and above. I think that is the success we have, that is achievement. That is very good. For the persons affected, it is a lifelong stigma, it's a lifelong burden. Unless the virus is taken out of the system, unless he is declared as cured, I don't think he or his family or anybody for that matter will be happy. That is why we have to move towards this cure, which is essential. Ever since this disease affected, I think all systems are doing their best to find a cure. Especially in India, we have a number of systems. Apart from the allopathy system, we have Ayurveda, Unani, homeopathy, Siddha, and a whole lot of traditional systems. Everybody is attempting something to find a cure. Sometimes we get information that vaccine is available, vaccine is coming. Someday we read newspaper that vaccine has been successful, major breakthrough is there. And next day, after 2-3 days, somebody will say, no, that is not correct. It will take another several years to find out a vaccine for this particular disease. Like that, sometimes we get good news, sometimes our happiness is thwarted, and we do not know what is happening in this vaccine field. That is one area I think honorable members may be interested to know from Saran. Usually people will say cure is not possible. Virus disease lifelong, because virus is ubiquitous in nature. It cannot be killed, whatever be the system, whatever be the drug. But when we are able to overcome a large number of viral diseases, how is it that HIV alone we are not able to go anywhere near cure? Here comes this important issue. It gets hidden in the system. It gets into the cells and hides and comes out at its convenience. So now Madam has found out a system how it enters the cell. With a lot of research experiments, they have found out how this virus enters into these latent cells. And once they can find out how it enters inside, I think they can find out a method how it comes out. That is her research, and I think she has done a lot of research, and we are all very eager to hear from her how this happens and how long we have to wait to see the good news that a cure has come out. I think you have the answer for it, and with your interest and involvement, I am very confident that you will give this happy news very soon to the world.
It's a great opportunity for us to hear Ma'am Sharon. We're all talking about preventing HIV and AIDS. But for me, this is the first time I'm sitting in a forum we are talking of finding a total solution for HIV and AIDS. That's the cure. We have been taking keen interest in finding relief to the affected people. We have been taking interest in seeing that it doesn't spread to the people. But today we are here to listen to Sharanji as to how we can cure this particular HIV and AIDS. A newborn child has all the resistance required, and that the child gets it from the mother's milk, the first day's milk or the first 3 days' milk. And an extract from the cows has been found out. But I cannot say how effective it is unless our researchers are going to tell us whether it has been able to contain the virus or otherwise. It is very essential for us to find a cure for HIV and AIDS. People with means, without means, the disease attacks, the immunity is destroyed. So it is very essential we are able to find a vaccine also. Research is on. We get solutions. We listen to news that we have been able to find a vaccine. But in India, we have not arrived at finding a vaccine. So far, the research activity is on. All of you are people engaged in this great task of finding a solution for HIV and AIDS. And from you, ma'am, if you can find a solution, it is that humanity will benefit out of this. And whatever the government of India or we, the members of parliament, can give support for your cause, that's our cause. We are fully with you. Wish you all the best. I'm happy that the Australian Commissioner is here. They have been engaged in this work for a long time, and whenever such issues come up, the Australian government has been taking keen interest in these things. I'm happy about it. And Subrajji has recently taken over, and the way he has been addressing issues, I'm very happy, and I'm sure we'll get the full support of our government. Thank you very much, and Jai Hind.
So I want to start by saying that anything I speak about with respect to the search for a cure for HIV should in no way come in front of our efforts to make sure that treatment is available for all and our efforts to prevent new infections. So the big question is, why do we need a cure for HIV? We heard earlier that we have outstanding treatments for people living with HIV. Treatment is simple, it's cheap, it's often just one tablet a day. It's now associated with very few side effects. Drug resistance is extremely rare. And we know that people who receive treatment for HIV have a normal life expectancy, the same as those without HIV. So why do we need to even think about a cure? Well, the first thing is that if you start treatment late, which is the case in many countries, here and elsewhere, life expectancy remains reduced. So not everyone enjoys normal life expectancy. And there is some ongoing morbidity on antiretroviral therapy, or what we call ART, with increasing frequency of diseases of aging and diseases such as cardiovascular disease and malignancy. So treatment is good, but it's not perfect. We also know that for every patient who initiates antiretroviral therapy, there are 2 new infections. So we're constantly going to be chasing our tails here in making treatment accessible to everyone. It doesn't mean we shouldn't try, but it means this is a very difficult avenue to pursue. And finally, I think funding lifelong ART for all who need it is unlikely to be sustainable. There's an estimate that antiretroviral costs will be close to $50 billion by the year 2030. So are we really going to be able to sustain this? It doesn't mean we shouldn't be trying, but it's something we need to think about for the long haul if we're going to, with the requirement for lifelong treatment for everyone. So when we think about cure, we think about the word means different things to different people. So I want to first of all explain, what do we actually mean by a cure? I think traditionally most of us think of a cure like we think of or with most infectious diseases. So we would need to eliminate all HIV infected cells. Measures of HIV in the blood, HIV RNA, would be less than 1 copy per mil. We wouldn't find it. This is commonly called a sterilising cure. It's how we think about curing most infectious diseases. This is probably going to be very, very difficult, and I think most people believe a sterilising cure is unlikely to be possible in HIV. What might be more realistic is to put HIV into remission. So think about HIV a bit like how we think of cancer. So long-term health in the absence of treatment. HIV RNA in blood is there, but at low levels, less than 50 copies per mil. And this is now commonly called functional cure. So what we're talking about here is a way of putting HIV into remission, allowing people to stop lifelong treatment and to remain healthy. So what we've learned over the last few years is that HIV cure is indeed possible. For many years people thought absolutely impossible. But about 5 years ago we really began to realise we were wrong, and there were a few key cases. I'm sure many of you have heard of these cases. The first was the Berlin patient, an American man, Timothy Brown, living in Berlin, who had HIV, was on antiretroviral therapy, received a bone marrow transplant from a donor who was naturally resistant to HIV infection. Timothy Brown stopped antiretroviral therapy shortly after the transplant, and the virus never came back. He's now 7 years off antiretroviral therapy, no detectable virus, a true sterilising cure, but the only case of sterilising cure we have currently. The next case was the Mississippi baby, reported early last year. It was a child born to an HIV positive mother who was not on antiretroviral therapy. The child was infected but received very early treatment 30 hours after delivery. The baby stopped antiretroviral therapy at 18 months, and the virus remained at very low levels and didn't come back. The baby now off treatment for 2 years. So example of a functional cure. And then we have heard about these interesting patients now called post-treatment controllers, or patients who receive treatment in acute infection, so early after infection, or in chronic infection more than 6 months after infection, stop their medicines and the virus doesn't come back. Again, very rare. The first reports were from France, the Visconti cohort, of now 18 patients off treatment for 8 years with virus at low levels, patients in remission, and very rarely this can happen if you start treatment in a chronic established infection. So HIV cure is possible, both sterilising cure and functional cure, although extremely rare. So what I'm going to talk about today is what are the major barriers to cure, why is curing HIV so difficult, what strategies are being tested, and some of the current and future challenges in this area of research. So I know many of you aren't scientists in the room, so I'm going to try and make the science accessible, but I want to explain to you the scientific reasons first of all about why, what are the major barriers to cure. So when we give people treatment for HIV, we know that HIV RNA, or measurements of virus in the blood, rapidly declines to what's considered undetectable, less than 50 copies or less than 20 copies per mil. However, if you stop antiretroviral, and at the same time a person's immune system recovers and CD4 T cells return to normal levels. But if you stop treatment, apart from the cases that I told you about, the Berlin patient, Mississippi patient, Visconti patients, in nearly everyone else, if you stop treatment, the virus comes straight back up, usually within 2 to 3 weeks. Now, if you measure other parts of the virus, so if you measure infected cells, and we can do this with a test called HIV DNA, you can always measure virus in a person on antiretroviral therapy. We don't see it in blood, but we can always see it inside cells. And if you use a very sensitive test of HIV RNA, you can detect it at very low levels. So although we tell patients you're undetectable, there is no such thing as an undetectable viral load. You can always find low levels of virus in any patient on antiretroviral therapy, but it stays under control. So the big question is, where is the virus hiding when a person has so-called undetectable virus? And we know from work from many investigators around the world that there are 3 reasons why HIV can persist on people on treatment. The first is something called latency, when the virus goes to sleep effectively inside a cell, and I'll talk about that in a bit more detail. The second is some low level of virus replication, even in the presence of antiretroviral therapy. And the third is the virus being able to get into some specific anatomical sites. So what do I mean about latency? And this is going to be the most scientific slide. So on the bottom are some cells that carry a virus, represented here in red, which is integrated into a person's HIV DNA. That's what's unique about HIV or any other retrovirus. It's able to become part of the host DNA, and therefore will persist for the length of time. It persists in a person for the length of time of the cell. Now, at any time, virus is still infectious. Virus can be released from this latently infected cell. The cell can be activated and release virus, but in a person who's on treatment, that virus can't go on to infect new cells. So the whole thing stays under control in a person on treatment, but there's always some infectious virus present. We also know that these cells can actually divide at something called homeostatic proliferation. And these cells are very long-lived. They're part of our immunological memory. So HIV has this unique ability to burrow inside our immunological memory, so we're stuck with it for life. The other way HIV can persist is that even when the virus is at very low levels, there can be continuing low-level replication. So antiretroviral therapy blocks most replication, but not all. And it's quite controversial whether replication exists, but I think we now have evidence that in about 1/3 of patients on antiretroviral therapy, there is low-level replication. It doesn't cause them any problems. It doesn't lead to drug resistance. A person wouldn't know whether they have it or they don't, but it's there in about 1/3 of people. And finally, HIV has the unique capacity to enter certain organs and infect certain long-lived cells in the brain, in the gastrointestinal tract, in the genitourinary tract, and in lymph nodes. And this also represents a way for the virus to sequester and persist for a long time. We don't know the true importance of these reservoirs. I don't think they're as important as long-lived, latently infected cells, but they're also a reservoir we need to think about. So the barriers to cure or eradicate HIV are quite formidable, but I'm going to tell you a little bit about what we're doing to try and address some of these ways HIV persists. And there are already a number of strategies that are being pursued, and I'm going to talk about the ones that are in clinical trial currently aimed at cure. And the first is to eliminate latency, these long-lived cells, part of our immunological memory that contain a bit or a whole piece of the virus inserted in the DNA. How can we get rid of these latently infected cells? And there's a few ways we think we can do it. The first is to treat very early during acute infection, like what happened to the Mississippi baby. The second is to activate latent infection, and I'll talk a little bit about work my own group's been doing in that. The third is to boost the immune response to HIV. And the fourth has been allogeneic stem cell transplantation or bone marrow transplantation, or what's happened since the Berlin patient. I'm going to go through each of these strategies and tell you a little about where we are with them. So first of all, treatment of early HIV infection. How might this work? Well, the idea is if you give someone treatment very early after they're infected, you might limit the number of latently infected cells if you get in there early enough. If you get in early enough, you'll prevent something called chronic immune activation, which is what persists in all people with HIV on treatment. And chronic immune activation means the virus is in an environment that favours its replication. And you can also, if you treat early enough, preserve HIV-specific immunity. And then together, that may give you a functional cure, which means undetectable HIV without antiretroviral therapy. What we know so far is that very early treatment certainly limits the number of latently infected cells. And I'm going to show you a few clinical trials which demonstrate this. And probably the best trial to study this is actually done in Thailand, where they've been able to recruit and find people very soon after infection and measure how much HIV persists in people when they receive early treatment. So on this graph, what you can see is how much virus persists in someone who receives treatment more than 100 days after exposure. And 2 years after treatment, you have about 10,000 per million, or 1 in 1,000 cells stays infected. This is the HIV DNA test I showed you, which is what we find in most patients. But in this study in Thailand, they managed to treat people, which is very difficult, but within 15 to 25 days of infection. They definitely had HIV before they received their antiretroviral therapy. 6 months later, even lower. And look at what you have at 24 months. Almost 2 logs less virus persists than if you delay treatment. And then they had this unique group where they actually found people in under 15 days after infection. Incredibly difficult logistically. Absolutely no way could you ever introduce this into a treatment program. But it shows you that if you treat really early, the virus can never get stuck into its hiding place. And so at 24 months, there's very low or undetectable levels in cells. Now, these patients haven't yet stopped treatment to prove that they're cured, but plans for those studies are underway and will probably be tested this year. But what we know is if you start treatment very early, you have very negligible virus that persists compared to if you start it after 100 days of infection. We see almost identical results in monkey studies. If you take a monkey and infect them with a relative of HIV called SIV, and you start treatment 40 days after infection, and treatment is shown here in a grey box. Like in humans, the virus rapidly disappears from blood, and like in humans, the virus persists as DNA or as infected cells. If you start monkeys on treatment within 10 days of infection, you can reduce how much virus is hanging around by a log. And if you start 7 days, just 3 days difference, you can have a profound effect on how much treatment persists. But what was very sobering about this work, just presented earlier this year in Boston, was that when they stopped the treatment in all these monkeys, the virus came back. So you probably need more than just getting very, very low levels of SIV DNA. You probably need a good immune system to keep whatever virus is left under control. So how often does it happen that you can stop treatment and with the virus under control? Well, I think it's pretty rare. I told you about the Mississippi baby, treated within 31 hours, had 80 months of treatment. Virus has stayed under control after stopping treatment. The French group have looked in their cohorts of people that start treatment early, usually within 2 to 3 months, and they estimate this happens in about 15% of people, but this is the highest estimate of any group. There are other groups in Switzerland who have looked at similar, when you start within 2 to 4 months, and probably only 9% can control virus after stopping. And a study done in Europe and Australia puts this at 5%. So for people that start very early treatment in acute infection, even at 2 to 3 months after infection, and stop treatment, maybe between 5 and 15% can control their virus. So this is why some people are saying functional cure may be possible for some patients lucky enough to get treated early. You may not have to be treated at 15 days, but if you're treated early, you've got a greater chance to keep your virus under control. The other way that we're trying to address this problem is by activating latent HIV. Now, this is a slightly counterintuitive. I'm sure many of you have heard about this approach, but for those that are not familiar with it, it's slightly counterintuitive because we spend our whole time with patients keeping the virus down and under control at the lowest levels. But the idea here is that you take a cell that's latently infected, so at rest, and has a piece of HIV inserted into the host DNA, and you stimulate it some way. You activate not the cell, but the virus. And you get the virus to start making copies of itself, start making the proteins, and start making new viruses. And this is all critical to generating an immune response. If the virus is just sitting in here latent, the immune response can't see any foreign virus on the surface of the cell. So stimulating the virus to expose itself to the immune system may work, and then that would lead to cell death. And the idea here is often called to shock or activate the latent cell and then lead to cell death or kill. And there's lots of drugs that can do this, at least in test tube models of latency, and they're all listed down here. And I've highlighted the drugs that have already moved into clinical trials, and they include the cancer drugs, which are called HDAC inhibitors, histone deacetylase inhibitors. I'll talk a bit more about those. Some cytokines called IL-7, and a drug called disulfiram, which is used to treat alcoholism. And clinical trials with these other agents hasn't yet started. So we did a study of an HDAC inhibitor, which is an anti-cancer drug called vorinostat. And I'm just showing you this schematic because this is how these trials are being done. Basically, patients doing very well on treatment for at least 3 years, low HIV RNA, normal T cell count greater than 500. They're on antiretroviral therapy throughout the entire study. That's critical because if you're going to activate latent HIV, you don't want any new virus particles to go on to infect new cells. So we took 20 patients who had been on treatment for many years, and we gave them this drug, vorinostat, which we thought would turn on latent HIV infection. Gave 14 days of drug, measured very frequently in the blood and as well in the gastrointestinal tract. And what did we find? Well, when we measured RNA inside the cell, we indeed saw that before treatment there was a significant increase in RNA on treatment, and this persisted off treatment. So we definitely woke up the virus. The cell-associated RNA is a way of telling how much virus is being made by a latent cell. But when we looked at DNA, or the number of infected cells, there was no change. So we woke up the virus, but we didn't eliminate any infected cells. And so there was no change in the viral reservoir. Maybe we didn't give treatment long enough. Maybe we need to do something in addition to activating the virus. And we also didn't see any change in RNA or virus in the plasma, which raised the question whether we were really producing virus. Now, since that study, another study has recently been reported by a Danish group in earlier this year, where they used a different HDAC inhibitor called panabinostat. They gave 4 courses of it, highlighted here in orange, and again they measured how much cell-associated RNA there was, and again they showed you could actually clearly activate the latent virus infection. And this time they showed that virus increased it in plasma, but again no change in the reservoir. So there is quite a lot of interest in these drugs that wake up the virus. We're sort of at the very beginning steps here. We've just shown we can wake up the virus, and there are many other studies being planned. There's another study of vorinostat dosing it slightly differently, which was recently reported to show no additional activity. There's a trial of giving vorinostat, the HDAC inhibitor, with a vaccine that will be given in patients during acute infection that will start in the UK later this year. There's a study of romidepsin, which is a more potent HDAC inhibitor, that will be done in the US this year, and romidepsin together with a vaccine. So a lot of clinical trials now happening, and we should hear the results of these clinical trials, I'd say early next year for some of them. And then there are drugs that are different to HDAC inhibitors. I mentioned disulfiram, which was used to treat alcoholism. In the first initial study done in the US, they saw a transient response or a transient increase in plasma RNA. And we're currently doing a study of high-dose disulfiram together with US investigators, and almost finished enrolling that trial. One of the big problems, though, with this approach is that latently infected cells are rare, about 1 in a million. So all of these interventions we're using actually aren't HIV specific. And a sobering example of this is vorinostat. Now, this is a complicated image, but this is something called a gene array, where you can look at thousands of genes at the one time. And each row, and the names here you can't see, represent different genes, and each column represents what happened in individual patients. So it's a way of looking at hundreds of thousands of genes at the same time. And we did these gene arrays on our patients before they received vorinostat and 2 months after they finished the study. And you can see here that if the colour is red, it means the gene's been upregulated. If the colour is blue, it's been downregulated. And what the striking thing that you can see here is a very distinct difference between gene expression in baseline. So this is each of the patients before they got the drug and 2 months after they got the drug. So we are causing big changes in gene expression. We're not just fiddling with HIV here, we're fiddling with many other genes. And this becomes a big issue around safety, because we're taking patients who are doing very well on treatment and giving them perhaps drugs that could make a very big difference long term. The other question is whether activating the cell alone will be enough, or whether you will need to do something else to kill it. And I think many people in the field think that what we're going to need is both activation and killing. So not just one intervention to activate the virus, we will need to shock and kill the virus. And there's a few ways we could come in to kill the cell, and that's with a vaccine or using an immunomodulator to boost the immune response. And this is a whole new area, again, initially pursued in cancer research that's being applied to people with HIV. And just to explain the principles of this, when you have HIV, most of your virus is recognized by T cells, but over time these T cells get exhausted and don't do the job properly. It's what happens with cancer as well. So you've got this foreign antigen there, cancer or virus, and the T cell stops doing its job properly, and there's ways we can tell it's exhausted because it expresses a molecule on its surface called PD-1. And PD-1 locks into something called PD-L1, and it's a way of saying stop doing anything, calm down, and you're now no longer going into action anymore. And people with HIV have characteristically exhausted T cells, which is why their immune system doesn't do a good job at getting rid of it. But there are now drugs that can block this interaction. They're called they can either block PD-1 or block PD-L1. And if you interfere with this, it revs up the T cell, or you get a reinvigorated T cell response. And they're now being used recently shown to be of great benefit in cancer, and in fact, unbelievably revolutionary results in melanoma, a cancer that's very common in my own country, and very, very effective because they give you these reinvigorated T cells that recognize the cancer. So a number of companies are making these drugs. One of them is Bristol Myers Squibb, and they recently took monkeys, infected the monkeys, gave the monkeys treatment, and then gave them 5 injections of this anti-PD-L1 drug and stopped their antiretrovirals. It was just reported a couple of months ago in Boston. And what did they find? Well, the monkeys that were given the control or placebo or isotype in red after their antivirals stopped the virus rapidly rebounded. The monkeys that got anti-PD-L1 in blue rebounded but kept the virus under control. So it did boost the immune response quite significantly. And more interestingly, when they looked at these monkeys that responded, they saw 2 groups. A group, 4 monkeys that had a really good response in keeping the virus under control, and 5 monkeys that were not as good but still better than placebo. So this could be the perfect thing to combine with an activating agent. And there's a study of anti-PD-L1 about to start in the US later this year. Finally, what about transplantation? Well, I started off telling you about Timothy Brown, photographed here, the only man that has been cured of HIV. And Timothy Brown was unique because he received a bone marrow that was CCR5 negative. You need CCR5 on the surface of your cells for the virus to infect you. And about 1% of Caucasians lack CCR5. So we don't know what cured Timothy Brown. We don't know whether it was the CCR5 negative bone marrow or all the stuff he had done in his transplantation. So many investigators are interested to know what part of the process led to his cure. Could we just do one thing and not give a full transplant to cure someone? So 2 years ago, there was some excitement about the Boston patients. They were men, 2 men with HIV on treatment who had lymphoma, a form of blood cancer, and they received radiation therapy, just like Timothy Brown, but this was a reduced intensity, and they received a bone marrow transplant from a normal donor, someone that was CCR5 positive. And the investigators followed these 2 men for 2 years and 4 years, and all their markers for HIV became negative. They were DNA negative and RNA negative. And back about a year ago, there was some excitement that perhaps these patients were indeed cured. So they went through the gold standard test to prove cure, stopped their treatment, did the virus stay under control? So they stopped their treatment and were measuring HIV in blood, in RNA, HIV DNA in cells, and it was looking pretty good at 12 weeks. And in fact, this data was presented in Malaysia last year. But at 12 weeks, suddenly the virus came back. So even though normally it takes 2 to 3 weeks for the virus to come back, these patients got a bit more virus-free time, but still at 12 weeks it came back. So this was very, very disappointing because they managed to get HIV DNA negative, but still there was some very low level of virus. And the same thing happened in the second patient, but this time he stayed undetectable for 32 weeks. But when the virus came back, it came back with force. Look at this, but over 2 days, the virus went from undetectable to over 1 million copies. So once the virus gets going, it gets going fast. So this has really raised the question of how low you need to go with any of these cure strategies. But being DNA negative wasn't enough. And my take on this is that you need both. You need to reduce the reservoir, but you also need a really good immune response ready to march into action with any virus that reappears. And finally, the last approach that's being used is making cells resistant to HIV. And this has been done in a very clever way through gene therapy, where we now have the capacity to literally use scissors to chop out genes. And this can happen, unfortunately genes aren't so easy to identify like this, but with the right sort of molecular tools, you can use these molecular scissors to eliminate a gene like CCR5 and make someone resistant to HIV. Or you can even use these gene scissors to chop out HIV itself. But the trials that have got going now are these gene therapy trials to eliminate CCR5. And this is how those studies are done. Patients that are HIV positive on treatment go in and have large volumes of blood collected. Then in the laboratory, those cells undergo these gene scissors to eliminate CCR5. And then the CCR5 negative or modified cells are expanded, so you no longer have any CCR5 positive cells, and then they're reinfused back into the patient. And that can be done with or without something called cyclophosphamide. And the results of this were reported just recently. And so this is what happened when they infused the cells back in. You had the total T cell count went up, the total CD4 count went up, but they also could measure these CCR5 modified cells, the gene modified cells, and they could show you got quite a few cells in there. About 20 to 30% of the CD4 T cells were gene modified. So that was exciting. First, gene therapies never really worked in any diseases as yet. So they showed that this was safe, that you could get the modified cells in, and they accounted for about 20 to 30% of the cells. But then they took the patients off treatment, and what happened Well, the normal CD4 T cells rapidly declined, as you'd expect, because HIV knocks off CD4 T cells. But the gene-modified cells hung in there. They declined a bit, but nowhere near as rapidly as the unmodified cells. So there was some survival advantage of these CCR5 modified cells. The trick now is to significantly boost the numbers. Now, gene therapy is high-tech, high-cost. Who knows if it will work? And if it does work, will it ever get to people, to low-income countries? So that's a big consideration when you think about how much to invest in gene therapy. But I think at the moment we want to find something that works, and then maybe something, a capacity to make it simpler. So just very quickly, what are some of the current and future challenges for HIV Cure? I think a big one is managing unrealistic expectations and changing our focus from what we know works. What works now is treatment, antiretroviral therapy, and the more people on treatment, the better. And that needs to be the top priority in the HIV response. Cure is really an important long-term strategy to hopefully avoid lifelong treatment. But, you know, we're not close to an HIV cure at the moment. We're many years away from it. We have some exciting new leads, but we're many years away from it. And it's really important that our patients get that message. But there's also many other challenges. I mentioned this earlier, we're giving interventions that at times have some risks and toxicities, and we're giving them to people who are doing well on treatment. And many patients still want to participate in these research studies. We have no shortage of people that want to participate, but we've got to be very careful here. This is not like treating someone with cancer who has a reduced life expectancy. We don't yet know. We need much better work to work out what will predict rebound. The Boston patients had negative HIV DNA for weeks, and yet they still rebounded. So we need better ways to find such low levels of virus, which we don't currently have. We need to, as I mentioned earlier, the expectations of study participants are key, and this can't get in the way of our commitment for universal access to antiretroviral therapy. There is a lot happening in this space. The International AIDS Society launched a strategy towards an HIV cure in 2012, and this has been a great tool for advocacy and making people aware, particularly governments and funders, about why this is important. And they've got the International AIDS Society has a whole lot of activities around trying to move the agenda forward. The Forum for Collaborative HIV Research, who are based in the US, are doing some great work on looking at many of the regulatory issues involved with HIV cure. They've got working groups with over 70 experts looking at these things I mentioned: biomarkers and endpoints, the risk-benefit of clinical trials, patient education, recruitment, and informed consent. You know, engagement of the community is going to be really, really important in this whole agenda. There's been a substantial increase in funding towards cure research. This is just some of the funders that have been very active in cure research. Without doubt, the biggest funder is the US government, the National Institutes of Health, and President Obama announced in World AIDS Day last year an additional $100 million towards cure research. So I think with some of the collaborative efforts happening, some of the funding efforts happening, we are going to see a big change in the science. And I do really believe that finding a cure is really too big for one laboratory or even one country. And what we need is collaborative networks. And we need those networks to involve low-income countries or countries like India that have huge burden of disease, have a track record in tremendous innovation. I mean, India invented generic drugs, and they would have so much to offer, and engagement of communities here in a place like India. And so at the moment, most of these funders, you can see US, Canadian, European, and I think over the next few years, what I'd like to see is greater engagement of many other countries. This could help everyone manage their burden of long-term antiretroviral therapy. So just in conclusion, I've shown you multiple but rare examples of cure which have been reported. They give both patients and scientists hope that a cure might be achievable. There's many strategies being tested in early proof of concept studies, like activating latency, gene therapy, immune modulation. It's unlikely that success will be achieved just by reducing the number of infected cells, and enhanced immune control is also likely to be needed. And finally, and I think we need to be very cognizant of this, that there is a significant additional challenge to find a strategy that's ultimately cheap, scalable, and widely available. A cure is not going to be a cure for a few rich Americans that can pay for gene therapy. And many other collaborators around the world who gave me different slides. Funding from the National Institutes of Health, the Australian Government, National Health and Medical Research Council, American Foundation for AIDS Research, and Merck. And it's in the final few slides. I just want to tell you a little bit about the conference. The title of the conference is "Stepping Up the slogan for the conference is "Stepping Up the Pace," to hopefully make people realise that we know what works currently to see an end to AIDS, or even maybe an end to HIV, but we've really got to step up what we know works and get it into places and people to really see the best benefit. Some of the key themes for AIDS 2014 will be around partnerships, that great advances in HIV have occurred because of partnerships between key affected populations, governments, scientists and clinicians, and we've got to maintain that partnership to see an end of AIDS through prevention programs, care and treatment for all. And I think that's first and foremost in our in the HIV response. Well, I think the stigma and discrimination is a big issue for us in 2014. We have seen some appalling recent discriminatory laws in many countries that discriminate against people most at risk of HIV, and that is a barrier to implementing what we know works. We've seen different speeds in the response of the epidemic, and a key theme is that no one is left behind. There are hotspots of infections in key affected populations, many of whom are here in India and here in the Asia Pacific region: men who have sex with men, people who inject drugs, sex workers, female transgender, are all experiencing increasing rates of infection. Many other parts of the world are seeing reduced rates of infection. How can we turn this around and make sure that no one gets left behind? The fact that the Millennium Development Goals, we will see new Millennium Development Goals post 2015 with a very big shift in HIV, in the attitude towards HIV/AIDS, and managing this transition is an argument for even stronger leadership in the future. And finally, the science. I'm co-chairing this meeting with Françoise Barré-Sinoussi, who won the Nobel Prize for discovering HIV. She is a basic scientist. She has been at the forefront for arguing and presenting the case for a cure. And both of us are very passionate about science making a big difference. And I don't just mean science as in new drugs and new fancy immunomodulators. I mean understanding the evidence of why something works or it doesn't work, and that's what we call operational research, and that's going to be critical in our HIV response, understanding or implementing what we know works. And of course the ultimate scientific goal for many of us is progress towards a vaccine and a cure. How are we tracking? We're doing well. We've received over 7,000 abstracts. I'm very pleased that we've had increased submissions from the Asia Pacific region. This region is often underrepresented at the International AIDS Conference, and it's marvellous that we're seeing great interest for people coming. We are on track to host 14,000 participants. If you look at the website, all the plenary speakers have announced, and we have someone living with HIV talking in every plenary session. And the City of Melbourne and the State of Victoria, Wayne will be pleased to hear, have gone crazy at the thought of 14,000 people visiting, and we have over 100 associated events planned for Melbourne to really raise awareness around HIV, and there's some wonderful things happening. The conference centre is right in the centre of the city, and so all of you will get a chance to enjoy Melbourne. Although it might be a little cold in July, it's still a fabulous city with lots to do. We've got a number of high-level speakers already confirmed. Many of you will, of course, know Tony Fauci, the head of National Institutes of AIDS and Infectious Diseases, Secretary of Health in the US, Kathleen Sebelius, Michelle Sidibe, your boss for many of you in the room, Helen Clark, the head of UNDP, Geneviève Fiorasso, the Minister of Higher Education and Research in France, of course our own Foreign Minister and our own Health Minister. And we've already had acceptance from Ministers of Health from France, Canada and Indonesia, and we're very we're looking forward to the elections being over in India. So because we would love to host the Minister of Health and Justice and Finance from India, and we will be making those invitations as soon as we know who will be in place for July. So hoping that many of you will come to visit us in Melbourne, and happy to take any questions. Thanks very much.
I actually have two very quick questions. One was in the Sobosubuvir trial, which came to give us a miraculous cure for hepatitis C. We've just discovered type 3, which is kind of very prevalent in our part of the world, has only 67% efficacy. And this is, of course, after 100% efficacy sort of thing had been announced recently. So I think that, like you say, the cure is a little bit of a magic bullet, and the steps towards a cure are sometimes more important than the cure in itself. And just linked to that, I watched a very interesting program by, I have to say, Sanjay Gupta, because that's how his name is pronounced, which talked about digital printing and the linkages of stem cell replication to digital printing, where in a way, sometime in the future, we could put our own bone marrow feeds in and get new, cells in a way generated to, and that in a way really brings us back to a reality. Like we know, Sobosubuvir doesn't cost very much to produce. It's the research and development, etc., which is the real sort of roadblock towards bringing something into public health domains. So in the technologies that are going towards the cure for HIV, are we getting these, let's say, bridal effects, where research and development, which is corporate owned or whatever, privately owned, is going to impair the accessibility of a cure more than we'd like it? Because we know that technology exists to make it quick, make it simple, and replicate it quite fast. The main thing is, how do we not just recover costs for what's spent, but ensure substantive profits to keep generating, to make it worth our while. To make it worth our while.
Yeah, so I think you raise a really excellent point about one of the great successes in HIV that has to be replicated in other diseases, and this is in hepatitis C. For those of you that don't know about hepatitis C, there are new treatments around, including a drug from Gilead called Sofosbuvir, with spectacular results, with cure with only 4 weeks of treatment in 100% of people with particular strains of Hep C. Not as effective in other strains which are more prevalent here in India. So, and astronomically expensive. So not a hope that it will be available to most of the people that need it, and in fact many, up to 30% of people with HIV have hepatitis C. But what the HIV response has taught us is that if you have the right partnerships and the right advocacy, you can change things like cost pricing, and that was done for antiretroviral therapy. And I hope that similar sort of partnerships with patients and similar advocacy and leadership will see the same in hepatitis C. And I think that is apart from some of the science that from HIV that has influenced other areas, I think that's one of the biggest legacies of the HIV response, the capacity to get together and advocate and change things. And Sofosbuvir is just a perfect example for what the community needs to do in the same way that the HIV community did. And I hope that that's something that will be discussed when we're in Australia. We are going to have a workshop before the main meeting on hepatitis C, because it's going to be linked to the World AIDS Conference. It's not just going to be doctors and scientists talking about new drugs. It's going to focus on issues that HIV has done so well, like drug pricing, like access to treatment, like rollout, like community-based prescribing, because all of that is completely relevant for hepatitis C. Digital printing and genetic makeup, I don't know much about that, but I guess what I'm hearing perhaps is that these things or interventions that sound so high-tech and so many years away may not be that complicated. And so therefore I think, and that's what we're seeing with gene therapy, and that we should probably still invest. And who knows what, how accessible that might be in 10 years' time or even less.
A real life situation from our national program in India. We have around 20 to 25 babies who were found to be DNA PCR positive after they were born to HIV positive mother. And then they underwent a whole blood testing for DNA PCR, and once they were confirmed, they were started on antiretroviral therapy. Now almost 20 to 25 babies at 18 months when we do antibody test, they are negative. So question is, do we take them as HIV negative? Do we stop their antiretroviral therapy? Because we are still continuing. Could this be potential functional cure? Though they were not started in 30 hours, they were started at varying time, varying from 6 weeks to 6 months.
Yeah.
So what should be the protocol once they are found to be antibody negative at 18 months?
Yeah. Well, I think the fact that they're antibody negative doesn't mean they're cured, because babies that haven't, when you don't for example, when you don't expose a baby to a virus for long enough, they never generate an antibody response. We will know if they're cured if you stop treatment and the virus didn't come back. And there was certainly new information present in Boston showing that if you treated babies in the first year after birth compared to later, there was a greater chance of being DNA negative. What we don't know, there's a high chance those babies are cured. We don't know whether you have to give treatment within 30 hours. We don't know what that window is. We just have one case where there was a cure. And I think what we're learning from many other reports are exactly as you described, children that were treated early remain antibody negative, as you might expect, but are actually DNA negative. And I think there are there certainly are investigators in the US who are now considering a treatment interruption study for these children. It would need to be done extremely carefully with regular testing and obviously consent from their parents, etc. So it's a tricky thing to do, but I think that's the only way we're going to have an answer. What may help though is a tremendous amount of work now going on into getting better tests than just DNA, because you saw with the Boston patients, they were DNA negative and the virus eventually came back, and they were being tested 3 times a week. So is that going to be feasible for your young children to be bleeding them 3 times a week? Probably not. So I think over the next few years we'll see better tests, and that may also help us in making some of these decisions. But there are clinical trials being done right now to see whether you can stop treatment in exactly the situation you're describing.
But presently in clinical situation we need to continue.
I would not, I think in a child this is experimental. It should, if it was going to be done, I would only do it in the context of a clinical trial. My personal view is I'd like to know more about the predictors of viral rebound when someone's DNA test is negative, and I would do those in that sort of test in adults first, just because of the complexity. But I think we might have some more answers in a few years' time.
What is the preventive measurement and the symptoms of the virus, HIV?
Prevention is still 100% a key part of the HIV response. I think we've known for many years about behavioural interventions with condom use, etc., preventing HIV, but I think one of the exciting developments in prevention have been around treatment as prevention, so that we...
90% prevention.
Yeah, so...
Full prevention.
Well, if someone that's on treatment, their infectiousness is reduced by 96%. And it may even be better than that, at least in heterosexual couples, what we learnt recently from the big partner study presented in Boston earlier this month. And all the other biomedical prevention interventions like circumcision, reducing the risk of acquisition by 70%, pre-exposure prophylaxis, giving uninfected people antiviral drugs. We haven't instituted PrEP at all in Australia yet. We're still struggling with how best to introduce that. And then topical microbicides or antiviral drugs that can be applied to the vaginal rectum to prevent prevention. So there's a real explosion in prevention technologies. And then the other exciting development is injectable antiretroviral drugs. So recently we heard about a monkey study where you could inject an antiretroviral once a month and protect the monkey from getting infected. So I think the whole prevention options have greatly expanded than what we once had, which was just condoms and clean needles. So the challenge for I think many countries is what's the most cost-effective biomedical intervention. Is it going to be treating everyone, treatment as prevention? Is it going to be PrEP? Is it going to be circumcision? And I think it varies in different countries with different epidemiology.
My question is to your slides on the acute infection in both the Swiss cohort and the German cohort. The success rate was rather low, 7%, 9%. Do we know why? And do we have any biomarkers on HLA system, chemokines, and other immune-associated genes that can help us find out these are the people who are going to benefit more than others?
Yeah,
because finding a HLA identical donor for transplant is also not a very easy thing.
Yeah. I think treatment during acute infection is not going to lead to functional cure for many people. The interesting group in France, the Visconti cohort, who are originally 14, now 18 patients who got treatment early and then stopped. They've been very well studied to try and work out why could they control their virus. And it looks like it's not their HLA type. It might be that they received 3 years of treatment and the other cohorts received 1 year or 2 years. We don't know, but I think what we need to do is now the French group have set up an international cohort of post-treatment controllers. We know these patients are rare. What we need to do is get them together and understand what were some of the factors that makes them unique. I think early treatment of HIV is really difficult. Not very few countries can do that well, and as most of you know, 50% of people are asymptomatic with acute infection anyway. So it's never going to be a realistic strategy. Why that's interesting is to use that as a tool to identify new research leads. I actually think the true figure is around 5 to 10%. It's very rare, and many of those patients may have been elite controllers anyway. That people that would have naturally controlled their virus even without treatment.
In India, the CCR5 negative in India is very, very low.
Very low, yeah.
Almost absent.
Dr. Bimal.
Yeah. So you nicely pointed the case. Yes, it is possible to cure, and it is possible to make the cheaper drug. But you have to do very early of infection. To do early of infection, you have to have like long surveillance program. So how is possible? What is feasible to 7 billion of people to another surveillance? Not in one time. You have to do multiple times. So how is that possible?
As I said earlier, I don't think treatment during acute infection is a pathway to a cure. I think it will identify people who can control their virus effectively after treatment, and then by studying those patients we will get clues about what might lead to a cure. I think changing surveillance programs to identify people very early, I don't think that's that's a strategy I'm talking about here. It's more the principle that if you get in early and you get the reservoir or number of infected cells very small, those patients may be best to test interventions.
Please.
You mentioned about the Berlin patient, and you discussed that it is considered as a functional cure, but not the sterilizing cure, despite having he is having no infection, no sign of infection for the last 7 years. So can you just tell me?
Yeah, I actually said the Berlin patient's the only example of a sterilizing cure. Yeah. Yes, I would call that a sterilizing cure. But I think that the distinction's probably not that helpful. What we really want here is a way to stop lifelong antiretroviral therapy. But I actually consider the Berlin patient a sterilising cure. Yeah.
There is also another baby that has recently, it was presented at CROI, the Long Beach, California, and the baby was immediately put on treatment because it was a high-risk case and was found to be positive at birth. So within 4 hours, the baby was put on. And there are other in Canada, in South Africa, so they're trying to get it. Just following up on what Dr. Newari is saying, because you need very high-end diagnostics, A, to get a testing at birth, and later on to see whether the virus indeed is not there. And you need a PBMCs to see whether the virus is there or not. So at your lab, is there any provision of if there is a cohort in the region which would need to be systematically followed up to see how long you need to treat a baby to consider a treatment interruption?
Thank you for bringing up the Long Beach baby. I don't think that, but I'm reserving judgment on that baby.
Only 9 months
And treatment hasn't been stopped, but received early treatment, and so therefore there was a lot of excitement, but I was less excited than most people because the treatment hasn't been stopped. You're right, to prove infection and start treatment very quickly, you need a lab that can have a very rapid turnaround. So that baby was treated within 4 hours, and they had 2 tests that were DNA positive. We would be very interested in collaborating on all of these assays of the reservoir up and running in my group in Melbourne. Would be really interested to collaborate. I should say that most of the assays are all optimised for subtype B. That doesn't mean that they couldn't be amended for subtypes prevalent in India, which is C, I think, predominantly. But obviously, what you would need to institute that early treatment is point of care testing, which I think will come for DNA as well.
I'd just like to say, Sharon, wow, it makes me proud to be a Melbourneian first and Australian second to hear you today, and it's just tremendous stuff, and it's lovely to sit in a room with people doing such good things. I had a prepared speech, but I'm not going to deliver it. I just want to say that our government, the Victorian government, which our capital city is Melbourne, some 4.5 million people, some 200,000 people of Indian origin, some of the best Indian restaurants in the world, is a very, very nice place to come for a medical conference. And I think that those who come down there, not only will they engage with some of the finest scientists in the world internationally, but also some of our fine Australian scientists. It's suffice to say that we have, I think, more than 11 teaching hospitals in Victoria, and I should say that my daughter's currently studying at one of those. She's fourth year, I should say, medicine, but she wants to be a surgeon, bless her. And we have a huge medical research precinct in the centre of the city, and we have wonderful hospitals like the Alfred, the Peter MacCallum, and Monash University, Melbourne University, all our universities working in this field. I think that Melbourne is a great place to have this conference, and we are hoping for all 14,000 people predicted to turn up in Melbourne from over 200 countries. But where I'm really interested is getting people down from India. Our government, the Victorian government's been in India since 2005, and for my sins, this is my second posting to India, which means I must love the place. And I live in lovely Bangalore, and I flew up this morning to be here today to hear Sharon and to encourage you, please think about it, talk to your colleagues, get your friends, get them down to AIDS 2014 to my wonderful city of Melbourne. It's kind of like Delhi and only totally different. It's a lovely place. It will be chilly, and for those who are interested in the snow, it's only 2 hours from Melbourne when the conference is over and you've finished that really good cerebral work that you're doing and the important stuff that you should be doing. I would really encourage you to come to Melbourne, and I think it's fair to say that Professor Sharon is just one example of the wonderful people you'll meet in our fine state of Victoria. So I encourage you to come, and thank you so much. And I know I'm in between high tea and a good chat among your colleagues who actually understand what Sharon's talking about. So thank you so much. And I did understand a little bit. So thank you, and please encourage your friends to come down to Melbourne for this wonderful conference. Thank you.